Thank you, Amy, for that wonderful introduction. And thanks everyone for joining. Let me share my screen and make sure it goes into presenter mode. And I have kind of a hodgepodge of topics that when I was putting this talk together, like most, probably most academics do this, where I wrote a title before I put the talk together. And I thought it would be fun to kind of cherry pick some of my favorite past experiments and research projects that will allow us to essentially hop across to some of the national parks around the world that I've worked in and highlight a few of some past experiences, but also talk about some fun projects moving forward. So a brief overview of the next 45 minutes, and depending on how quickly I talk or how much my dinner food coma sets in, I might go closer to that 45 minutes than what I'm planning. So I'll start by giving you a little overview of my path to where I am today. And this is primarily for the students and younger people in the crowd that might be interested in hearing about some of the unique and weird and fun experiences that I've had that have led me to my position that I have today. I'll also then introduce us to this idea of what it's like for plants to ride the escalator to extinction, as it's termed, and I'll describe that a little bit later. And then our first national park that we'll visit is San Pedro Martir National Park in Mexico. And I'll spend most of my time talking about this project, and I'll explain some of the details, but primarily it's because I'll focus most of my time on that because the California Botanical Society funded a large portion of that project. So this is kind of like a progress report or a final report, sort of. We'll then jump across the Pacific Ocean to Daisetsu-san National Park in Hokkaido, Japan, and we'll talk about plants from a slightly different angle, going from native species to invasive species and how climate change and some of our actions are influencing different types of species that we care about or don't care about, but we should. And then we'll jump back to the US, we'll jump back to North America, and we'll visit Saguaro National Park, where a lot of my work takes place today. And I'll tell you about some work that I finished there, and I'll hint at some of the work that I'm still pursuing there. And then I'll give you a brief glimpse, it might be a rush glimpse depending on how much time I have left, of some of my current projects with the US Geological Survey. So my path, as Amy mentioned, I got my bachelor's degree at New York University. I studied biological anthropology, and I am a native New Yorker. I grew up in New York, and all through my undergrad, my experiences with nature were pretty limited, and my understanding of the natural environment was pretty limited to what I got in textbooks and then what I got from some fun but not so serious field projects working in Central Park, and then daydreaming about what my future career might look like as a biological anthropologist, potentially studying long-tailed macaques in Southeast Asia. I'm not sure, but those dreams quickly changed after I graduated and moved to the Rocky Mountain Biological Laboratory in Goffin, Colorado, where I was hired to sort of do a pseudo REU experience. And I was studying burying beetles and climate change. And it was my first encounter with true wilderness. And while I was there, I took a few field courses, one in like field methods in ecology. And I realized that although macaques were cool and studying beetles were cool, I really liked plants because they didn't move and they were slightly easier to collect data on. And I also realized that studying plants gave me like the best excuse to be outside and hike in beautiful areas like this here. So from that turning point after my undergraduate degree and working at the Rocky Mountain Biolab, I knew I wanted to sort of shift what the focus of my resume was to make me a plant expert so I could be outside. And the best way to do that was, for me at least, working for various federal agencies as an intern or in some cases a paid technician, which is pretty rare. And I spent a few years after my time in Colorado working for the National Wildlife Refuges and the Bureau of Land Management. And I was primarily doing rare plant surveys, some vegetation mapping, some fire effects monitoring, a little bit of invasive species removal, but I was trying to get as much field experience as I could to, I don't know, explore what the careers might be. At this point, I don't think I knew what my future career would even look like. I just knew that I liked being outside and I liked when they paid me to be outside. After my time at the Fish and Wildlife Service and in the BLM, I then moved to the National Park Service, where I spent a little over a year working for the Sonoran Desert Network, which is part of the Park Service's inventory and monitoring program. And it was there that I kind of first understood that humans played a much larger role in the management of our lands than I had previously thought when I was just an intern at the other agencies. And it was the Park Service that my supervisor and my mentees at the time really encouraged me to go back to school and pursue a graduate degree if I was going to try to stay in the federal government as a resource manager or somebody working to understand some of the impacts on our parks and other protected areas. So I went back to school and got my master's at UC Merced. And sorry for this long autobiography. I hope it's beneficial to at least some of the students that are watching. So during my master's at UC Merced, I was studying the impacts of elevated temperature and drying soils on alpine plants in Colorado, which you can see in some of these pictures. We have these infrared heating arrays that are literally warming the side of this mountain. And there I am counting grasses or something and measuring responses to the treatments that we were exposing them to. You'd think that after all of that and with my master's degree, I'd be ready to go back to the Park Service, but I realized that getting paid to ask my own science questions while being outside and designing my own experiments was a lot of fun, so much so that I followed up with what some people would say is pretty sadistic, and I went and got a PhD. And I got my PhD at UC Irvine, and it was during my PhD that I really, at least at the beginning, I struggled to figure out whether I could be a sort of jack of all trades researcher like I've always wanted to be, or if I really needed to focus in on one very specific niche. And because I had a great advisor, Travis Huxman, who you'll see in a couple slides, I was encouraged and allowed to pursue as many interests as I was capable of tackling without sacrificing, the intellectual merit of my dissertation. So I ended up studying a combination of both desert plants, like I had been doing for the federal government in those years as an intern and technician, and then also continuing to pursue my passion of working at high altitudes and studying alpine plants. And after that, where I am now is I'm a federal employee. I'm a research ecologist with the US Geological Survey. And I consider myself a climate change ecologist, a part-time plant ecophysiologist. But overall, I would really label myself as an evolutionary ecologist because I really strive to include evolutionary principles in our understanding of how plants work and function on our public lands. And I've been working for the last few years trying to convince the managers that I interact with and work with on a regular basis that protecting plants in our parks and monuments isn't a matter of just monitoring and recording vegetation cover, but understanding the evolutionary drivers that are pushing them to where they are today, and also what they're going to look like in future times. So kind of a vague theme that I want to keep throughout this talk is the idea of boundaries. And I'll start with my project in Mexico, where we kind of designed an experiment that was getting at this question of whether plants are moving on up, and then also moving on up in terms of elevation and how warming is pushing their distributions and ranges into higher elevations as the climate warms. But also what this means for species that don't potentially have the chance of moving up if they already exist at the highest possible elevations possible. So I put together just this slide that includes a few academic articles over the years that I've collected that have kind of quirky ways of describing this elevational shift that's expected. And the stories never really, the framework doesn't really change from study to study. The conclusions and findings usually do, where maybe some species or morphological types are moving at higher elevations, but others aren't, maybe because they're being outcompeted or the niche space is changing. But I just like the way that if you follow just titles in academic literature, you can learn a lot about a field, whether it is all uphill from here, whether there is a disproportional risk of habitat loss at higher altitudes because of the limited available space that plants at the highest altitudes can move up to, and also whether there's this escalator effect that as warming continues, whether plants are just going to be marching up. That's kind of a big question, and there's a lot of data to support that that's the case, and that there are species-specific responses. But I don't know whether it's healthy for us as the human species to create grim titles like this, but I still love it, where it's, you know, plants are riding the escalator to extinction, as depressing as that sounds. And as an evolutionary ecologist, the way I think about it is, well, you know, we have our mountain ranges and we have our happy little sunflowers, and maybe they'll just migrate upwards until they can't anymore. And then maybe they'll be outcompeted or the climate will change again. Maybe we'll lose them from part of their range, but they'll be maintained in a distant part of their range, in a different range. Or maybe they'll stay where they are and move to the tops of the peaks and kind of adjust their life history strategies if possible to deal with the new environment that they're experiencing. But maybe they can't possibly get as big as they can in this picture, in part because they're probably going to be outcompeted by the other species that are moving uphill behind them. Maybe they won't change their appearance to us above ground, and instead they'll change their resources below ground and how they strategize to deal with the novel environment that they're experiencing. I'm not sure what it is, but I'm hoping that the next portion of the talk that you'll see sheds the light on why I think understanding phenotypic plasticity, or how variable plant traits are within a population, within a maternal lineage, how understanding that is kind of a nice step towards predicting and better understanding how alpine species and other species we care about are probably going to respond to environmental change. So we'll visit our first national Mexico, which is San Pedro Martir National Park. And it's, let me just go back, it's special to me because the Annetta Carter Memorial Fund funded a portion of this work. And when I applied to it, I think it was in maybe 2013 or 2014, I knew about this park. I knew about a handful of species that were there. And I kind of wrote this proposal to meet the deadline in the hopes that somebody would be like, yeah, we need more science to happen in this park, because I know that a majority of what happens in the park is restricted to astronomical observations because there's a fantastic astronomy observatory there. And there's also a ton of work, at least in the plant world, related to forestry topics like fire ecology because of the unique history of the park that you'll hear about in a little bit. So I wrote this little proposal, and then I worked with some colleagues who really helped me refine some of the ideas and expand them as I was learning about the park and the study system that I was interested in. And Michelle Lynn, who's in the top left here, she was an undergraduate at the time, and we hired her to work in the lab, and she ended up developing an honors thesis project related to some of the work that I had done in the park. And she won an award. She presented right before she graduated. And she really carried out a lot of the work that you'll see in the next few slides. So a brief overview of the park, just some key details that I think are fun to know. The park was established in 1947, and it's the 4th largest national park in Mexico. And for a summit junkie like me, it's exciting because it contains the highest point in Baja, which is Picacho del Diablo, which is the mountain in the picture on the lower left. It's also the southern end of the peninsular range, which extends up into the United States. It's a special place floristically because it has a really high level of endemism. There are tons of high altitude species that you can find there and nowhere else in the world, in part because of the Sky Island that really is the national park there. Some people describe it as a botanical wonderland, and I totally support that. And in this picture, I borrowed from Michael Kaufman that it is a special place for the California floristic province because it is, at least from the perspective of conifers, it's the southern terminus of range of 5 really important conifer species. And just within the park, which, as I mentioned, it's huge, from the lowest elevations, it's coastal sage scrub, and then it quickly transitions into chaparral, and then up into Jeffrey pine, mixed conifer. There's some alpine or subalpine meadows, depending on how you want to define them. And then where most of my work took place, which is on the alpine rock outcroppings that are on the highest points. The park has a unique history in that it's never been logged, which is just an insane thing that this exists, and then more people aren't talking about it because you get this really intact system with old growth forest that's like overwhelmingly beautiful when you're there. And there's been little management activity in terms of human impacts. There's been a limited fire suppression that began in the 1970s, and it is tied a little bit to some of the grazing that's happened in the park in the past. So there are about 500 species in the high country, and I think about 66 of them are endemic to the Martyr. And there have been a few climate change studies, modeling studies that have focused on the Baja Peninsula. And similar to what we would expect from almost anywhere now, there is a prediction that there will be increasing temperatures and that longer, more intense drought events are likely to occur in the park. So I wrote this proposal for the Annetta Carta Fund just to study this species you see here, which is Stephaneria monocephala. And I picked it because when I was looking at Annetta Carter's background and what work she had done in Baja, I ended up somehow connecting to all of the great botanists that have worked in Baja. And one of them was Reed Moran. And he described these 12 new dicots in 1969. It was published after a 1968 trip that he had done with a large group. I think it was partially led by the California Botanical Society. Anyway, so he took these 3 pictures in 1968 of Stephaneria. And at the time, he thought he was the first person to describe the species when he ended up publishing this. That turned out not to be true, but I did write the proposal with the idea that I would go to the park just to study this species. And after I received notice that I was going to be funded for the project, I immediately dove into as much literature as I could find about the park and different plant species, mostly with the concern that I wouldn't be able to identify the species once I got there, or if it was missing, would I be able to find something that was equally as interesting that would make a valuable use of my time. And I want to just point out that the San Diego Natural History Museum has this fantastic resource, which are all of the field notes, or most of the field notes of Reed Moran, including most of his trips throughout Baja California. So I downloaded all of these images and I read through all of his journals, especially the entries for his trip to the Martyr, so I could better understand just what I should expect. And what I learned by reading through his notes and his natural history observations is that the idea that I should just focus on Stephanomerium monocephala may have been short-sighted in when I wrote the proposal, and that there were probably more species that I should consider because they're co-occurring species. And when you find Stephanomerium monocephala, or the few times that people had seen the species in the past, it frequently occurred with these species with sometimes similar morphologies that were living on these rocky outcroppings in these crevices, and were also endemic and restricted to these unique habitats. So I decided that I was going to be prepared to go study all of the species at the park. Not all of them, but as many as I could that were in this unique microclimate habitat on these rocky outcroppings. So I got my hands on as many of the type specimens or photos of the type specimens as I could, just again, because I had this paranoia in my head that I wouldn't be able to identify any of the species once I got there. I also had really poor Spanish language skills. There were also no botanists that I could convince to come with me to the park because of scheduling conflicts. I also spent a day at the San Diego Natural History Museum going through their herbaria collections, taking pictures. I think I took hundreds, 200 to 300 photos of as many specimens as I could that I thought I might encounter at the park, just so I could be prepared. And I brought most of those photos with me. I probably looked like a crazy person. So ultimately, what happened during this field trip is I ended up collecting field observations for the 6 species that are listed here. Again, with the goal of going there solely for Stephanomeria, which I have now many, many more observations than we last had in 1969, when the last observations were made, though some people have documented species occurrences since then. And I bolded the text for the last species, Heterotheca brandegeei, because that species ended up becoming the focus of most of the work, simply because of phenological timing. So we got there, and for some of the species, it was just a little bit too early in the year to collect high quality phenology data, or what we were really after, which were seeds. But Heterotheca was fruiting and seeding at the time of most of our observations. So we were able to collect seeds and bring them back to the lab, where Michelle ended up doing this fantastic growth chamber experiment. So a little bit about Heterotheca. It's a mat-forming rhizomatous perennial. It has wind dispersed achenes, and it's a beautiful, beautiful charismatic species when you see it up close. It was first described in 1896 as Chrysopsis brandegeei. It was named after Brandegee, who originally collected it in 1893. I think the first specimen was collected. And then Reed Moran accidentally redescribed it in 1969, not knowing that it had already been described, but that's fine. And then it was reclassified in 1988 to the species name it has today. And as of Reed Moran's observations in specimen collections in 1969, it was known from only 6 sites in the park. So we went there with the intention of absolutely revisiting those 6 sites and hoping to find it elsewhere as well. And I thought this was a great species as I was like working through the park and collecting data, that this was a great species to answer this questions related to escalators to extinction, and if this was the type of species because there was no altitude above it for it to move to in terms of habitat. Also that it's this wind dispersed species, so it has like potentially these interactions among the different populations on the different outcroppings in the park that might lend itself to some interesting questions related to climate change impacts. Oh, here are just a couple more fun things that I love as a historical nerd. The original description from, I think it was 1896, and also the original type specimen that was collected in 1893. And then just a snippet of Reed Moran's description, which here it's in Latin, from 1969. And I'll just mention that this work has already been published. It was published in 2019. We published it in Conservation Physiology. And it's kind of it's like a tome of a paper. And it's one of papers I've written that I'm quite proud of, because I really got a chance to include all of the unique natural history tidbits in the paper that I've been forced to take out of other papers because, you know, for whatever various academic, political reasons, whatever they are. So you could check out this paper, and I'll present some of those findings in the next few slides. So our goals were to complete a reconnaissance to determine the status of high elevation endemics as we proposed to the Botanical, California Botanical Society when I originally wrote the proposal. But then with this new focus on heterotheca, we wanted to ask questions like, is there enough phenotypic variation to buffer some of their populations from things like drought and warming that are predicted, especially during the first growing season of this plant's life cycle, because we know that establishment, especially in high altitudes, is already a challenging thing. And when you add into the mix changing climates, I think that becomes even more challenging, as you'll see in some of our results. We also want to ask whether the species is able to adjust its allocation strategies depending on the stress, or whether it's temperature, whether it's drought, with the expectation based on previous studies like the movin on up studies that I showed before, that we would probably see an increase in below ground growth, and maybe if the species is capable of it, which it probably is because it's master, that it would be able to utilize a higher water use efficiency when we were putting it through its various stress tests. So we revisited the 6 known locations, and then we also carried out a transect survey across the outcroppings on the crest of the Martyr, which was about 20 kilometers or so. We also collected seeds of heterotheca from 16 separate maternal lineages, and we carried out trials to estimate germination rates, because we didn't know anything about this species other than that it probably shares some similar characteristics to other heterothecas and asters that we have some data on. We, and then I mean mostly Michelle, the undergraduate at the time, carried out this great 120-day growth chamber experiment where we subjected individuals to a few different treatments, being just what we thought were ambient average July through September conditions, a drought treatment that it performed essentially the most severe drought scenario for some of the climate modeling papers we cited. And then also the most extreme warming scenario for the Baja Peninsula, the Martyr in particular, which was about a 4-degree C warming, and then an interaction of the two. We measured kind of a standard suite of traits to get a response to these climate treatments, including above and below ground biomass. We counted the number of leaves, which was kind of tedious for some of these plants because they form these really, really dense rosettes with sometimes hundreds and hundreds of leaves. We calculated specific area on every single leaf. And we measured leaf carbon content and water use efficiency by using stable carbon isotopes. And we designed this experiment with a mixed model approach to examine treatment effects. But then for me, I was most interested in getting at whether we could partition some of the variance that was explained by things like sites where we collected the seeds, or maternal lines that we collected the seeds from, or just random variation across individuals. So these red dots on this map here show all of the sites along the crest where we were able to locate Heterotheca. And there were a couple other sites that aren't listed here, but we didn't include them because there were like maybe 1 or 2 plants, or we weren't able to collect seeds from them at the time. So they weren't included in our analysis. So we first had to do germination trials to see if we could even get the seeds to work for us in the experiment. And we weren't sure if they were the type of alpine species that they produce seeds and then they overwinter, and that breaks some type of dormancy, even though they're an aster, and then they would germinate in early spring, or if it would be an early summer thing, like right when the seeds are dispersing, or if it would be more of a late summer thing once the monsoons peak in the region, important part of the ecosystem there. And what we found through some greenhouse and growth chamber experiments is that they're most likely germinating in the wild during the late summer at the peak monsoon, which just so happened to mimic our greenhouse conditions at the time. So we created this germination curve that you see here on this graph, showing how long it takes for them to germinate on average, and what that relationship looks like. We then exposed these plants to these treatments, and what we found was some shifts in how they were allocating carbon, in part because they were just so severely stressed. So we saw a 60% decrease in above ground biomass in our drought treatment, and a 55 to 60%, 66% decline in below ground biomass in both the drought and the warming plus drought treatment. And I'll use my cursor here to just show you that what you're seeing in these bars is each bar is the total biomass of the plants averaged for the treatment. And then the white portion of the bar, or the hollow portion of the bar, is just the above ground biomass. And the below ground biomass is represented by the color part of the bar that you see. Individuals in our warming and drought treatments produced fewer leaves, 54 to 72% fewer leaves across the board. So we were definitely, our stress treatments were working. But interestingly, there was no detectable change in the quality of the leaves and the leaf carbon content, or the specific leaf area, though you might see a general increase here with level of stress, if you want to call it. However, we did detect a pretty dramatic decline in water use efficiency in our warming and drought treatment, which probably suggests it was just like a level of stress beyond what they were capable of dealing with. Most depressingly, though, because this was, an early life history exploration, and we only were looking at the first 120 days of growth, the survivorship through time was pretty dramatic across the different treatments, where we saw only about 19% mortality under ambient conditions and 54 to 65% mortality in our warming and drought treatments. The nerdier stuff for me is that what we see in this random effects breakdown to understand the influence of individual locations where we collected seeds, individual maternal lines that we collected the seeds from, how variable individuals are. What we learn is that there's still a lot of variance that we can't explain, that we just don't understand because the system is pretty new to us. But that between individuals within maternal lines, there are some influences, just natural variability across the individuals changes what their strategy is in response to the treatments. But then more interestingly, I think, is that between maternal lines within a given location, we're seeing some influence on how they change the content of the carbon content of their leaves or their water use efficiency, which might suggest that there's like a strong influence of the individual microclimates that those maternal lines were exposed to. And I'll admit that we don't know if this is genetically controlled or if it's just environmental. We only looked at one generation, But there are still some interesting things that we could probably imply from these results, that reduced biomass and fewer leaves might serve to reduce water stress in the species. Also, that maintaining a high specific leaf area and similar carbon concentrations suggests that there's at least temporary compensation when they're experiencing these stressors. But that microhabitat differences experienced by the maternal lines may also play a role, as I mentioned. And also primarily that reduced water use efficiency suggests that the warming and drought that they're likely going to experience and probably already are might be too much to handle. And that maternal influence explains the ability of plants to adjust their water use efficiency. But we really need to figure out what's controlling this and how plastic it might be or not. So some future directions while we end this park and jump to the next few for a few minutes. And I'd love to write a proposal to go back to the park with anyone who's interested. I'm really curious to find out, well, of the community of plants that we collected a little bit of data on, including Heterotheca, who's facilitating who, if at all? And do these species interactions influence how they're going to respond to stress and future climate change? We also need additional surveys. There are so many places of the park that I just wasn't able to go to because I didn't have time, or I didn't have climbing equipment because I'm not a technical climber. And there are outcroppings where these species probably occur, but we just don't know because nobody's gone there, or we haven't gone there and recorded that information. I'm really also interested in estimating gene flow across these little alpine islands that they exist on and how they're interacting with each other. And also, I'm really curious to find out if some of the species that we recorded data on are clonal, in that they're expanding their root systems through these cracks and fissures in the boulders and popping out at different ends. So potentially when you're counting the number of plants on a given boulder outcrop, it might be one individual, as some previous studies have shown, but haven't been done in this particular system. And before I quickly jump to a few other parks around the globe, I want to especially thank the California Botanical Society and the Annetta Carter Fund, who really gave me the opportunity to go there, and UC Mexis as well, which really made the collaboration across the border possible. And there are tons of people that were involved that have like a behind the scenes role that made this work happen. So now let's switch gears from native plants that are living at the precipice and we know barely anything about them to an invasive species that is displacing a ton of native species that we know a lot about and people care about, but don't necessarily know what to do with this invasive plant. And this work, I need to acknowledge the funders and also a few collaborators, including Gaku Kudo, who is in the top right here, who really, he hosted me when I worked with him in Japan. He hosted me in his lab, and he really introduced me to this amazing study system that has a really interesting history of interactions with humans, and then also our interest in conserving the alpine species that they care about in these parks. And similar to my discussion about the escalator to extinction, and my interest in boundaries, this tree line boundary is really interesting to me because it's a hot topic in climate change biology. And this idea that there's this imaginary line that's driven by environmental drivers, but then also plant niche space and how they're able to establish and persist depending on where this line is, and how where a lot of people are working to predict how this line is going to shift with climate change, if it's going to be equal across the board. And I can't help but think of Julie Andrews, and if she'd be singing about how alive the hills are with the sound of music as our alpine wildflower habitats change to something that's more of a darker green and maybe shrubby or filled with trees. And we've published a couple of papers from this study system that I'll put here just so you could jot them down quickly. One related to some of the strategies that this invasive plant is probably employing to allow it to be so successful. And then one about what we might be able to do if we're ever going to be able to fight the species and restore the native the native wildflowers that are protected in this park. So the species that I'm going to talk about for just a couple minutes, and I'll go quickly through the slides, is this Japanese dwarf bamboo, Sasa curulensis, and it's invasive, and it's invasive for a number of reasons that are clear. It's a clonal species. It vegetatively reproduces. It produces these amazing monocultures that can be up to 10 hectares that are just one single clone. And we don't know about its ability to acclimate, but we know that it's hardy because the culms of the plant live for 3 to 4 years below the snowpack. And as the snow is melting, they're already photosynthesizing even before the snow is fully removed from them. And they're doing their thing to invest more and store as many resources as they can below ground. So it's weedy. So I say watch out for the trees and shrubs that Julie Andrews might care about, but maybe we should also be watching out for the grasses. So we worked in the system along this elevation gradient where we studied the species in its native range. So it's a native invasive in this park. It's native to the montane forest understory. And only in the last 50 or 60 years or so has it been encroaching into subalpine meadows and also into alpine meadows above tree line. And we did part of our study was looking at aerial imagery, and there aren't many numbers here other than you could see that the colors represent Sasa curulensis, Sasa, in the different elevation bands we were interested in studying. And this is a image of the species in 1977, color-coded by elevation range, and then in 2012. So this is kind of like a magic eye trick where you kind of just need to squint and take a peek or dig into the paper if you want to see some actual numbers. But you can see that the species is expanding into open spaces, and that's why we're able to detect it with these unique color bands. And in particular, that there's been quite a bit of activity across its elevation range, but especially at the subalpine tree line and into the alpine zone, which is demarcated between this, primarily between this transition between this greenish color to this bluish color. And what we found by tracking the species growth through time is that the species is capable of adjusting its growth strategies depending on the environment that it's in, which is kind of unsurprising, but or intuitive, and that it's related to how it interacts with available water and its water use efficiency strategies. So as you move up in elevation from the forested site in green to the alpine site in red, you see an increase in water use efficiency, but a decrease in the relative growth rate. So this here is total relative growth rate of all of the individual components of growth, the culms and the leaves that we measured through time. And that a lot of the changes we saw were at least correlated to a seasonal dry down rate. So how quickly the soils were drying at a given site influenced how the species was able to shift its allocation strategies to be able to invade into the alpine and subalpine meadows. So we saw in Mexico that there's this native species that has a ton of, at least some phenotypic variation that allows it to respond to environmental stressors. And we see in Japan, there's this invasive species that also has this flexible morphology that is allowing it to move up an elevation to become more alpine-like. And it seems stressed like most alpine plants might appear on first glance, but that there's this physiological compensation that might be promoting its invasion, and that it's related to a soil moisture dry down. And just to save time, I'm going to quickly go through a study on Saguaro National Park because I want to give everyone in the audience a chance to ask a couple questions. But I first want to slow down on our Thursday evening. Well, hopefully some of you are having a glass of wine or tea, and realize that the slowness of growth and development in the cacti teaches a lesson of patience, which has incalculable value in our lives of contagious haste. And I love this quote, and I love its representation of the saguaro growth rate for a variety of reasons. And it's one of the reasons why I'd like to continue working with saguaros for the rest of my life if I have the opportunity to do so. So I was recruited to, not recruited, but I was invited to come up with a study to help shed some light on how saguaros responded to climate change. And it was in part spurred by some social media and other press organizations getting a hold of a before and after picture of part of the cactus forest at Saguaro National Park, starting in 1935 with this picture here, and everybody freaking out that the saguaro was going to go extinct because of this picture in 1998. But if they had talked to park resources or resource managers or biologists, they would have learned that there's a lot happening in these two pictures that you can't understand at first glimpse, in part because there's probably population turnover happening, and a lot of the saguaros that are on the right aren't even visible in this photo because they're growing beneath their nurses or their shrub trees, their nurses, shrubs and trees that are an important part of their growth cycle and how their population's established. So we published this work a few years ago now, and what's cool about it, and I'll talk very quickly, is that we have data, estimated ages for something like 30,000 individual saguaros that was all collected by citizen scientists and volunteers working in the park over the course of years. So we were able to, just by measuring height at a few points in time, calculate growth rates and relate them to ages, so how we could estimate when the plant's established, essentially. And what we came up with was this graph, number of saguaros establishing per hectare across time, at least since 1950. And what we found with very little effort was that there was this really strong relationship with the Palmer Drought Severity Index, which includes a metric of temperature stress, but also drought stress. And it seems like, just looking at this correlation graph that you're looking at here, that populations of saguaro are regenerating in the park based on how bad the drought stress is in a given year. And what we found that's a little bit more nuanced is that depending on where you are in the park, you might have that'll really determine what chances you have for your subpopulation in reestablishing. And if you're in the low elevation sandy Bajadas and it's a pretty bad drought year, you're probably not going to see much recruitment. Same thing in the mid elevation foothills that are slightly rockier or less sandier. But then once you're up at the steepest slopes that are really rocky, that's where we find these probably nice microclimate pockets that are allowing the saguaros to establish and persist at young ages because of potential water retention or overland flow that's allowing them to do their thing. What we're looking at now, and that I hope to share with this group in a year or two, is insights into the nurse-protege relationship that the saguaro is famous for, particularly trying to understand where saguaros get their water from, because they have incredibly shallow root systems. And we have some preliminary data, and we're exploring it further, that suggests that saguaros may or may not have parasitic tendencies or are opportunistic scavengers where they're seeking water from their their nurses that they're growing under or near, and are potentially receiving a water benefit from them. So in the very last few minutes, I'm going to quickly tell you that maybe this will spur some questions. I'm really interested in boundaries, both ecologically speaking, whether it's Heterotheca at the uppermost limit of its range and what that means for it to not have a boundary to move into. But then also the dwarf bamboo we saw in Japan and how it's moving beyond its original boundaries into new habitat that it's invading and displacing native species that are really cherished there. Or saguaros, and depending on where you are on the landscape, whether drought might play a severe role in how their populations are going to respond to change, or less of a role if they're in these really relatively happy microclimates. So at USGS, I think about these boundaries a lot, in part because I most of my projects are on the Colorado Plateau, which is represented here. And it's just a land management bonanza of sorts where you have almost every federal agency and tribal lands that are all abutting each other, and there's relatively little private land. So there are tons of opportunities for managers to interact, and hopefully scientists like myself to be able to provide answers and ideas about issues related to boundaries. That just because you might be from a different federal agency, the fence that separates your land from another federal agency's land doesn't necessarily mean very much to the plants that are moving across that boundary. So 75% of the land is managed by federal agencies or tribal agencies, and there's tons of human pressures on it in terms of visitors to parks and other public lands, energy development, grazing, invasive species, you name it. So I am working a lot with the Genetics for Western Restoration and Conservation Research Group at USGS. And we primarily use molecular genetics to understand populations and how they span boundaries and environmental gradients. But we're also really focused in on developing an understanding for our partners that will allow conservation ideals to be carried out moving forward, and species that we care about to be protected moving forward. And one thing that I'm really focused on right now is when we collect seeds and we bring them out onto the landscape for restoration, how do we identify what the right seed is and where is the right place to put it and when is the right time to put it out on a landscape, especially in our drylands. And the last thing I'll briefly mention is some work I just started on a different type of boundary. So it's a totally human, social, political boundary. And it's our southernmost border of the continental US with Mexico, where if you see in this map, and I'm sure you've heard just in the news over the last few years, this region has gotten a lot of attention because, in part because of how many wild and protected conservation spaces there are. So this map on the top is color-coded by different federal agencies that manage the lands that abut the border. And I'm really interested in understanding how these different management agencies are sharing knowledge with each other, how they go about restoration when there are human impacts like building a border fence or illegal traffic and foot and vehicle traffic moving through parks that degrade desert habitats and take many, many years for biocrust communities and other soil communities to reestablish. So these are just some of the questions, and I hope that this very quick overview of some of the work that I've done in some of these parks and protected spaces has been somewhat insightful, and I'll happily take questions if there are time before everybody falls asleep. And with that, I'll thank you, and I'm happy to expand or touch base about any of the things you've heard here, including if it's just students who want to know about my academic background and how I got to where I am.